So let me start out with an old sort of AI paradox, which is attributed to Hans Moravec, where basically this is back in the 80s, where there was a big surge of interest in AI, and basically what he said was, whatever we find hard will be easy for computers. And this is evidenced by, for example, if you want to multiply 16-digit numbers or find the square root of 2 or something like that, computers can do this much faster than us. But actually what we find easy, the tasks that we actually find easy are going to be the hard ones. And those are the ones that are actually going to be hard for computers. And so in today's world, this is still true, because we still don't have computer algorithms that actually can see. We don't have algorithms that still hear and recognize speech. We don't have we don't have robots that really move around and interact well in the world. They're getting there. There's been good improvements, but still they're nowhere close to where humans are. So the question is, what is missing? What is it that we are doing so well? Of course, one answer is that the things that we do without thinking are actually stuff that we've optimized over the course of evolutionary timescales, and so they're actually optimized, and therefore all the solutions, all the processing that is happening is behind our awareness, and therefore we're not aware of all the difficulty of the problem. Okay, so many of you are probably interested in both computation and to some extent neuroscience. So I just want to put out a little bit of reasons of why we should be having some kind of dialogue between AI and neuroscience. This is obvious, actually. This has always been happening, even with the early days of AI. So what is the appeal here? What is the appeal of actually trying to connect brains and AI? Well, two things in my mind. One is that you can understand brains using AI. So for example, AI algorithms that are better may match with the brain better, and then that leads to insights. There could be things using AI algorithms that you can do without, which you can never do with the brain. So for example, I can take a pre-trained deep neural network, tweak every possible connection that I wanted, and I can tweak the learning algorithm. I can do a lot of things that basically you can't do with a brain. So that's one sort of nice thing, and actually this is an active area of research today. Many researchers are actually trying to take brain data, trying to match it with AI algorithms and seeing how they match up. The flip side of it is, of course, we can improve AI using brains, and so, for example, insights into how the brain works may lead to improved AI algorithms, and that's really the appeal of AI and neuroscience. And I believe, and I think many of us would believe, that such a dialogue is possible. It's not very easy, because it requires actually having expertise on both sides, or at least being able to appreciate or talk the language of both sides, and that's not very easy. But certainly it's a fruitful area, and we hope that insights can come on both sides. Okay, so let's again go back a little bit into the history. This is actually a book that I was very inspired by Douglas Hofstadter, and basically this is attributed to him. Douglas Hofstadter basically talks about this. He says that there are two hard problems of AI. The two hard problems are, what is A and what is I? Okay, and for all of you, since you've come to the great land of India, we know that there is no I, because the Buddha said so, and so really the only problem to solve is what is A. Okay, and actually it's very interesting. Douglas Hofstadter, back in the day, in the 80s, talks about this problem, and basically he shows this graphic. This is directly from his book, and he says that, look, these are all images which you would easily and regularly recognize as the letter A. But it's quite possible that many of these particular patterns of letter A you've never seen before. You've never seen the A written in that style, and yet all of us would understand that these are all letters, the letter A. And so somehow we have understood this concept of A-ness, and we are able to operationalize it, and we are able to recognize the letter A across many fonts and many appearances. But this is not true for AI algorithms today. For example, you train an AI algorithm on a bunch of fonts, the algorithm will recognize A in that particular font or the fonts that it's been trained with, and then start to make errors the moment you go away from the trained data. So the question is, how is it that we are able to generalize so well because we understood something about ANS, and somehow the computer cannot, or we are not able to make computers understand what is ANS. Okay, so yeah.
The general thing is valid, but there is a cheat in here, right? Because you have a very strong context when you look at this image from
Of course. I would say that.
If you presented me that triangle down the bottom right,
sure.
I would have said, hey, but
sure.
now I will have to do it.
That's right. The context, of course, helps, because I'm telling you that all of these are letter A. But I would say that we are very robust to distortions of the letter A, and that point still stands. That in fact, you're able to recognize the letter A across huge variations, which machines basically can't tolerate even today. So the question is, what is it that is making our vision so good? Okay, and so let's go into the brain. This is the monkey brain, which I'll talk about later also today. And it turns out that monkeys have very similar visual systems to us. They have very similar visual processing. And what's also shown is like a variety of different brain areas that are there in the monkey brain, which are dedicated and are involved in processing vision. It turns out that vision is actually not easy for the brain either. Just like computers struggle with vision, the brain also sort of struggles with vision, in the sense that there's a lot of real estate which is dedicated to visual information processing in the brain. And so if you think about the area occupied by the retina in your body, it's a very tiny fraction, but the area occupied by vision in your brain is huge, much larger than the proportion of the body occupied by the retina. And so this is an indication of the fact that visual information processing is also a hard problem for the brain. It's not like the brain has some magical solution and then there's just some very little processing, or the eyes have something magical and you do something magical and you get it. The brain also has to do a lot of processing to get there. So that brings us that brings me to kind of give you an overview of what our lab does. This is our lab at ISC. I often show this picture to everyone because it's still for us daily proof that computers are still not actually very good at recognizing letters. And this is why you see those tests on websites today where you have to fill out and recognize distorted letters. And that's because by and large, computer algorithms are still not there. Even on the simplest tasks, we don't really see them completely there. They're really getting better, but still they're not completely there. It's still a big performance gap. So how do we actually understand how vision works in the brain? Well, you've got to do some experiments to figure out how vision works in the brain, and you've got to unpack all the complex processing that the brain is doing. So here's one set of experiments that we do in our lab have to do with studying human participants. So we bring people into the lab, or nowadays we do these as online experiments. You can run an experiment online, collect some behavioral responses from people. You could also take them into an MRI scanner where you can actually look at the brain activity evoked by images or when they're solving a task. More recently, we've been looking at reading in children to understand how or what makes a child learn to read fast. We're also looking at perception in autism and ADHD, and these are all ongoing studies which I won't be talking about. What I will be also talking about is, if you want to understand the visual system and the complex information processing in the brain, you've got to also go into the individual visual areas and try to understand what's happening in the visual pathways. And so a major chunk of our lab is also involved with running experiments in the monkey brain, where we're recording from regions which are important for perception and recognition. And then we're trying to understand the format of the representations in those areas of the brain. We're looking at behavior in monkeys which are performing tasks. They're trained on complex tasks, they perform it. We're looking at perception in monkeys, which I won't be talking about, and we're also looking at natural behaviors, which I will be talking about. So that's the overall sort of scope of work that we do. So there are two parts to my talk. In one part, I'm going to talk to you about vision in brains and machines, which is probably a topic of more interest to all of you. In the second part, I'll talk to you about how we're trying to understand the neural basis of real-world vision. And this is more recent work, which is probably not too relevant to this workshop, but then something that we are very excited about, and I think will have an interesting impact on how we understand vision in general. Okay, so let's go to the first part. And let me dive into one sort of very basic question. This is a general question really about any visual system. So you can say, well, I have a visual system I want to study, and the question is, does that visual system see the way we do? Okay, so what do we mean by that? What does it even mean to ask that kind of question? Well, a lot of comparisons that we see between brains and deep networks have to do with looking at some kind of correlation between the brain activity, and then also you have some kind of activation in the deep network, and there's some correlation being performed. And what I would say is that we are often seeing results like this, which are actually quantitative differences between brains and machines, in the sense that there is some benchmark score, and then you say, okay, the brain score is this much, and then the algorithm score is this much, and the algorithm score is usually less than the brain, or the algorithm is not able to completely explain the brain data, and so you're getting some kind of score which is actually a quantitative comparison. What is missing in this quantitative comparison, what is missing with these kinds of comparisons is that these are actually not telling us any properties which are different between brains and deep networks, or brains and any other visual system or a computer vision algorithm. Okay, so how do we actually think about properties that could be there in brains but not in deep networks, or vice versa? So what I would say is that those are actually qualitative comparisons. This is all work done, by the way, by Georgian, who was a recent PhD student from the lab, Harish, who was a postdoc, and Pramod, who was also a PhD student. So basically what all of us decided to do was to think about whether we can test deep networks for various kinds of properties which are present or already known to be present in brains. And so by compiling a list of properties, you could say, okay, the deep network actually has this property, or perhaps the deep network does not have this property. And for example, if you find that deep networks have this property but not as strongly as the brain, well, maybe you can improve it by adding more data, or tweaking the architecture. But then if you find that the deep network does not have the property, or has even the opposite trend, then you can't fix it by just simple tweaks of the architecture or data set and so on. So you have to do something fundamentally different. That is at least our intuition. So that's how we went about it. So we wanted to say, okay, are there any qualitative properties based on which we can say, okay, the deep networks are actually seeing the way we do, or experience the visual world the way we do. So that's the basic setup. So what do I mean by qualitative comparisons or qualitative properties? One good example, and this is what actually the entire field of vision science and psychology has actually grown because of establishing a lot of these properties. So for example, there's one property which is very famous. This is the Thatcher illusion. If you look at these two faces, they look almost identical to you. This is the inverted face of Margaret Thatcher, the British Prime Minister. Turns out that if you actually put these faces upright, then you'll see a very big difference between them. So you can see that, right? So when the faces are upright, you see a very strong difference between them. The same two faces, if you put them inverted, you don't see a difference between them. So the question is, why is this happening? But the fact is that this is a property that is actually integral to your vision. And what this really means is, what we realized is that we can't ask a deep network whether it recognizes Margaret Thatcher, but we can ask the deep network whether it differentiates between two faces. And so what this means is that in our perception, that the upright faces are actually far away from each other. That is, you can differentiate upright faces very well. You cannot differentiate inverted faces very well. What this really means is that the Thatcher effect really implies that the distance between upright faces is actually more than the distance between inverted faces, despite the fact that the image is exactly the same. Okay, so somehow, because obviously we're trained on looking at upright faces, so obviously this is the case. But now this becomes a nice way to check whether actually deep networks, which are actually trained on various kinds of object categories, maybe they've never seen faces, do they acquire a Thatcher effect? So these are kind of interesting questions you can ask about deep networks, or for that matter, any visual system. So the question is, do deep networks show the Thatcher effect? The question is very simple. I can measure the distance between two upright faces in any layer of a deep network. So you take the activations produced by a particular layer, a vector of activations for the upright face, vector of activation for another upright face, and you compute the distance between the vectors. So if you calculate the distance between the vectors, I don't know if there's a pointer here, but anyway. So if you look at the distance between the vectors, yeah.
But this assumes that the dimensions of measurement are same, right?
That's right. So it does not assume that, because what you can do is to compute a scale-free index. So what you can do is you take a distance between upright faces, take a distance between the inverted faces. All we are checking is whether the upright face distance is actually larger than the inverted face distance. We don't care about the units of the distance, and actually, like all good neuroscientists, you can compute an index. And so you have an index that you can compute, and then you can say, okay, this is a scale-free measure, and a positive value of the index means that it's human-like, right? So this is exactly what you can do. So you take the deep network, you have the feature vector you can extract from the faces. And then basically what's interesting now about the deep network is that because it has layers, you can actually compute whether it has a Thatcher effect at every layer of the deep network. And so what you then get is a kind of, great, thank you. So what you can then get is a kind of map of how the Thatcher effect is present in the early layers or not. And then you can see how that map sort of evolves across layers. So this is what we do. So here's the Thatcher effect. And so 0 and above means that it's that you have a Thatcher effect. If it's a negative value, it means that it doesn't have the Thatcher effect. In fact, it's more sensitive to inverted faces compared to upright faces. So if you plot this now for human perception, if you plot this now for a standard VGG network, this is a standard VGG architecture which is trained on ImageNet. So the ImageNet database, by the way, does not contain people, or it contains people or faces, but they are not labeled. Okay, and turns out that this one actually shows a kind of negative value here, which means that it's kind of more sensitive to inverted faces. But if you take a deep network, which is trained on upright face recognition, the same architecture, but now trained on faces, then you see a pretty robust robust Thatcher effect. What this means is that the Thatcher effect is actually emerging in the later layers of the deep network, and it's not present in the early layers of the deep network. So this is also giving us some interesting info that how much perhaps computation might be required to get an effect. So if, for example, an effect emerges only in the later layers, perhaps it only emerges in the later layers of the deep network, and maybe it's a clue that it might also emerge later layers in the brain as well. So these are interesting comparisons that you can start to make when you make these comparisons, when you make these kinds of effects. Okay, so this is one, the Thatcher effect. What we then decided to do is to test a number of such properties. Let me show you one more and I'll show you the full list. The next property that we wanted to test was something called global advantage. This is a very famous effect reported in the 1970s by David Namon. And basically the effect is like this. If I show you this shape and ask you to report whether you see a circle or a diamond, a circle or square, for example, here, you're going to be able to very quickly report that this actually is a circle at the global level. And you'll only take a little longer time to report that this contains squares or the circle is made of squares. So the point here is that humans actually report global shape faster than local shape. This has given rise to the idea that we see the forest before we see the trees. So that is, you see the global structure of a scene before you see the local details. Okay, so now the question is, can we convert this into a statement about the distances in feature space? Because if you can convert this into a statement about distances in feature space, we can check those distances in deep networks. So that's what we decided to do. And what we thought, or what we reasoned, was that if humans are able to report global shape faster than local shape, maybe it's because they can notice a global change faster than they can notice a local change. So if you perform, for example, if you make humans perform a visual search task, where they're searching for this as target among those as distractors, or that as target among those as distractors, people actually notice the global change much faster, or people actually search for the global change much faster. So we are able to show that this is actually an effect that is there in the distances in feature space. And so now you can actually then convert that into an index again. And so what you can say is, well, if a visual system is more sensitive to changes in the global shape compared to local shape, it has a global advantage. This is exactly like what humans have. Now the question is, you can query any other visual system and say, okay, let me compute the feature distances, compute the index, check whether global sensitivity is there or local sensitivity is there. So here we find something very interesting and different, which is that if you take a standard deep network, or actually we tested this across many deep networks, all of them were feed-forward deep networks trained for object recognition. More recently, we've tested the same property for self-supervised learned deep networks, and as well as recurrent deep networks and various kinds of architectures. All of them have the same property, which is that they show a local advantage. Okay, and so that what this is indicating, that humans are actually very sensitive to changes in the overall shape, but deep networks are actually very sensitive to changes in the local shape. They're not sensitive to changes in the global shape. Okay, this has been also confirmed by very similar such experiments that were done in deep networks like style transfer, texture transfer and all that. All of those are consistent with the fact that deep networks are picking up very local features and not really understanding the global context or the overall shape of an object. Okay. We tested this, we applied this approach to a number of different properties. This is the full list of properties that we tested in that study. And it turns out that deep networks do have a bunch of properties which are similar to humans, but they also don't have a bunch of properties which are there in humans, which deep networks don't have. So what we are getting now as a result of this is a kind of a checklist saying that these are all properties that deep networks anyway have. These are properties that they don't have, and now this might pave the way to actually being able to train deep networks to have these properties and potentially get more human-like performance in deep networks. Okay, so this is one sort of fruitful way, at least in our minds, to be able to compare brains and machines and be able to come up with some list of qualitative differences that brains and machines might have. Okay, so this is the first part. So if you're comparing machine and human perception, this can reveal sort of qualitative differences between them. The next question is, of course, can we investigate vision at the level of single neurons? And this study is going to be going back to that sort of old, the one I showed at the beginning with the CAPTCHAs. Okay, so the question is, can we have a fruitful sort of interesting comparison between how the visual system is actually representing distorted letters, and then can we compare that with how deep networks or other vision systems are representing distorted letters? Okay, so here's the experiment. We have a monkey which is sitting in front of a screen, and this is just a picture of the brain. We're recording from the inferior temporal cortex, which is a region which is known to be important for recognition. So if you have lesions in this region, animals experience severe deficits in recognizing objects. If people have stroke in these parts of the brain, they also experience agnosias, which are basically disorders of perception, where your vision is okay, but you're not able to recognize objects. So these are actually important regions that are crucial for perception, for normal perception. What we're doing in the experiment is actually to insert an electrode into the brain. The electrode is insulated all the way till the tip, so it's picking up signals, and it's picking up electrical signals. These are extracellular recordings. And on a good day of recording, you'll get a signal like this, where you have a nice neuron which is isolated close to the electrode. On a bad day of recording, it just looks horrible and it's torture, but you'll know it only if you've been there. What we can then do now is to actually, I'm going to replay to you an experiment that was done here at ISC, actually not too far from here. We are at the primate research laboratory, which is sort of a walk away. But of course, you can't get in because there's all kinds of regulations about getting in, so don't try to sneak in, okay? The experiment is the following. You're going to see what the animal saw. The animal is actually trained on a task to just look at images, and just for looking it gets reward, okay? So even here in this experiment, you're going to look at images, and just like the animal, you'll get a juice reward or some food reward later, but it's not going to be immediately after the trial. The animal gets a juice reward for just looking at the images. If he looks away, there's no juice. If he looks continuously at a series of images, he gets juice reward. What I'm going to play now is a video of the recording where you're going to see what the animal saw, and you're going to hear over the speakers the activity of a single neuron like this converted into an audio signal, okay? You're going to hear things like trrr, trrr, which are actually not noise. Those are actually the most beautiful signals known to mankind, which is the sound of action potentials coming from the brain. And it's a great feeling because you're actually eavesdropping on the brain at work. You're listening to the activity of the brain, and you're then being able to correlate that with what the animal is actually doing real time. So here's the task. Okay, so all of you probably noticed some correlation between the audio and the video. And basically, this is if you figure, this is the holy grail for us, because this is a region that responds very uniquely to certain objects but not others. You might have noticed here that these neurons are responding to all the animal images, but also responds to the fish, also responds to that hand drill. So it's not really uniquely to like an animal, it's not an animal detecting neuron. If you keep recording from neurons in this region, you'll see neurons that respond to hand drills, they'll respond to projectors, fans, all kinds of shapes, and according to principles that we don't really understand. Someone who figures out the principles of coding in this region will become really famous, because that representation is what is key to object recognition and key to making vision work in machines. You might have also noticed something else, which is that the neuron responds the same way to two views of the object. Okay, the exact same response or almost the same response you get for multiple views of the same object. And if you think about it, this is the kind of neural representation you need to be able to recognize an object, because you don't care about which view you're looking at, you just need to be able to recognize the object. So if you want to recognize your friend's face, you don't care about whether they're looking towards you, away from you, or whatever, or they're close to you, far away from you, and these neurons actually do that particular function. Okay? Yeah?
Has the monkey seen these images before?
No. No.
These are all new images?
These are all new images, and actually we see pretty nice view invariance for objects that the animal I'm pretty sure never saw. For example, we had images of motorcycles, trumpets and things like that, which we're damn sure that the animal never saw, unless he's sneaking out of a facility and kind of going to jazz clubs. Okay, but basically we see pretty much similar kinds of invariance invariance for novel objects as well as objects that are kind of familiar to the animal. Yeah.
Are you sure that for the obvious for which the neuron didn't respond, the monkey wasn't looking away?
Yes, because we have eye trackers looking at the animals, which are monitoring the gaze of the animal, so we know where exactly he's looking. So we know he's looking at the images. So, and if you keep recording from different neurons in the brain, you will see neurons that respond to the objects that you're showing. I mean, this is only one example neuron. Think of millions of neurons in this region, and across those millions of neurons, you'll see neurons that respond to a particular object. Okay, and a given neuron responds to multiple objects, and a given object activates multiple neurons. So let's not think of it that as a kind of very unique kind of, it's a many-to-one mapping on both sides. Yeah. Okay, so coming back to the question of distorted letters, well, what makes our brain so good at reading distorted letters, right? Because if you think about this experiment, we're going to be showing these distorted letters to a monkey, and the monkey has never learned to read. And so the question is, why do we even think that monkeys would be able to actually be able to represent or have a good representation of distorted letters? Well, I would say that the brain is actually quite used to actually experiencing distorted objects. And so if you look at these images that were shown in the experiment, you can see here that the dog actually got pretty nicely distorted in another view. And so if you, when an object rotates in 3D, a bunch of features go out of view, bunch of features come into view, some features get compressed, some features get expanded. So it's not a trivial transformation at all. And so the fact that your brain is able to handle a viewpoint change already shows that it's able to recognize objects across certain distortions. And what I would argue is that perhaps the kind of distortions that you're experiencing with distorted letters are the kinds of distortions that you're anyway used to doing. And therefore you have a pretty nice object representation system which will represent both familiar objects, unfamiliar objects, and even letters for you. Well, anyway, this is all speculation. We got to prove it. What I'm going to show you now is the recorded activity of many neurons responding to these distorted letters. And so you can actually see that those neurons actually respond pretty nicely to distorted letters. You can extract the identity of individual letters from the CAPTCHA and so on and so forth. Okay. This is all work done by Harish, who is a postdoc in the lab now at NIH. And what Harish and I decided to do was to ask, can we extract some principles from neurons which are responding to distorted letter strings? So we looked for two kinds of principles or two kinds of patterns. The first question is, how do neurons respond to distortions of a particular letter? And so what we said is, let's consider two strings, and I'm presenting the same string in two possible distortions. So the string is the same, but you're distorting in a certain way, exactly like you do with a CAPTCHA. But very importantly, we took the same string, we took two different strings and applied the same distortion to both of them. So the idea was that you're kind of creating a combinatorial matrix where you have strings on one axis and distortions on the other axis. And the question we wanted to know is that whether you could separate the string information from the distortion information. And because if you're able to separate the string information from the distortions, that means you're separating sort of styles from substance, and you're going to be able to recognize the letters well. That was our idea. Okay, so we can test explicitly for all of these things, because you can then say, can I take this response matrix and see whether it's separable or not? The other question that we also could ask is by taking and querying the neurons for the response to combinations of letters. So the question is, if you take a neuron that responds to, if you take the neural response to a combination of letters, can I explain that in terms of the response to the individual letters? And is it a sum of the individual letter responses? Is it a product of the individual responses? Or maybe there's a neuron that responds to a particular unique combination of letters, and maybe it doesn't respond to other combinations of letters. Okay, so this is all sort of the setup. Yeah, question?
I just have a question or a comment. So maybe you know this experiment of this newborn chicks that were exposed to some images,
right.
And within their first days of life, they
right.
were able to kind of like recognize images given a viewpoint, but it was not that good. So they kind of learned that.
So some of those are imprinting experiments where actually the chicks would respond in a very particular way to a predatory bird kind of silhouette or things like that. Those are mostly in terms of there's some sort of automatic patterns that especially lower mammals or other animals actually have. We don't have any of those innate responses. We don't seem to have those innate sort of sudden, or sort of innate responses like that. Yeah.
What I want to say is that if you train your network to be invariant of an object rotation, it has to learn that.
Yeah.
That's the thing. but it's not inherently. If you just train an analyst and then you
Yeah, so I guess let me answer it in the way that what I would say is that we don't know if the view invariance that is learned by the animal is a result of the visual experience the animal had, or because of genetically sort of encoded some sort of feature extraction or whatever. All we know is that at this point, when we're recording from the representation in the neurons in this region, it is already view invariant, and it doesn't seem to depend upon prior experience with that object, because we are taking novel objects and still we're getting pretty good invariance. Of course, I should qualify that by saying that when people have tried to take objects and familiarize that to the animal, there is a subtle increase in the viewpoint invariance, but the effect is not very large. Most of the effect is already there. Yeah.
How are you quantifying the responses, neural responses?
So you heard that burst of action potentials produced by the neuron. Sometimes you get a burst which contains lots of action potentials, and sometimes the burst contains no action potentials. So what we do is to quantify the firing rate of the neuron in terms of number of action potentials in a small window of time. And that's all we're doing. It's basically looking at rate coding, but even that we are not able to still fully understand the variability in the neural response as a function of the image. Okay, yeah. Okay, so this is the basic setup. So we want to understand how neurons respond to distorted strings and how do they respond to multiple letters. Okay, so here's an example response. What I'm showing you here is there's a set of letters being presented to a particular neuron. The letters are either distorted according to a fisheye transformation or a rotation or a global distortion or whatever, and these are the responses. On the x-axis is position, so we're showing the same letter at multiple positions relative to the center of gaze. Remember, in vision everything is relative to the center of gaze. Okay, move your gaze and the entire visual field is changed. Okay, and what is shown on the right hand side, this is a predicted response, assuming that the information about shape, which is the letter identity, and the distortion are actually multiplied together. Okay, and what we can then say is, can I compare a couple of models where the neural response to a letter at a particular distortion is the sum of the letter information and the distortion information, or is it a product of the letter information and the distortion information? Okay, and so this is the result. What I'm plotting here is for each dot as a neuron. On the x-axis is the reliability of firing of the neuron, because some neurons are more reliable, some neurons are less reliable. And then on the y-axis is the model correlation for the additive model and the multiplicative model. And basically what you see is a very subtle increase in the model correlation for the multiplicative model. Okay, and this is a small increase because if you think about the product of two numbers and the sum of two numbers, they're going to be highly correlated. So there's only a small set of range where the sum and the product can be distinguished from each other. So the difference in the model correlation is a very tiny model correlation. But if you take these kinds of neurons and you simulate neurons which add information versus multiply information, it turns out that neurons that multiply information can decode information much better. So if you train a classifier on neurons that multiply letter information with distorted information, the decoding of letter identity is much better. Decoding of distortion is also much better, and this is true if you multiply two signals. And so this is one reason what we think is that if you multiply two signals, it's actually better for decoding in this particular context. Okay, so that's one insight. So what we are finding is that the response to distorted letters is a product but not the sum of the individual shape and distortion tuning. Okay, so let's look at multiple letters, and so I'm showing you the same sort of layout as before, but now I'm showing you actually the responses of a neuron to multiple strings. These are 2-letter strings, 3-letter strings, 4-letter strings, and so on, across multiple distortions. And the question we wanted to know is whether the neural response to multiple letters can be explained in terms of the individual letter responses. So we have the individual letter responses and we also have the string responses, and the question is what is the relationship between them? Turns out here that we actually get the opposite pattern, which is that the additive model is actually better. And if you actually instantiate this in a simulated population of neurons, again the additive model is actually better. So if you want to decode individual letters if you have multiple letters, the better way to encode them is to add them together. Okay, whereas if you have a letter which is distorted, then you need to take a letter and then multiply it by the distortion. Okay, so that's sort of two rules that seem to come out of this analysis, and these are all small differences. So the question is, does that really matter? Is it really important? And that's what I'm going to address right now. So one rule that we found, or this is the rule that we find from the neural population, is that the response to distorted letters is a product but not sum of the shape tuning and distorted tuning. Okay, the response to multiple letters is a sum but not product. So there's a nice kind of double dissociation that we're getting. Very subtle effect, but it's there. Okay, so now the question is, are these two rules sufficient to crack CAPTCHAs? Because in a recorded population of neurons, there's a lot of heterogeneity, and neurons could be doing some complex linear summation, nonlinear summation, all kinds of things. So what we decided to do is to then do a simulation where we take neurons which embody these rules, and we took every possible combination of the rule that we could think of. So here's the logic. I take neurons that could be sharply tuned or broadly tuned to letters. I take their response to shape, position and distortion to be the sum or the product. I can take the response to strings as the sum or the product. And then finally I can apply or not apply divisive normalization, which is a rule that we know applies in visual cortex. So now we say for each of these populations of neurons, for every one of these combinations, I have a population of neurons. I can take that population of neurons and say, can I decode the letter identity in a CAPTCHA? That is basically, can I read the CAPTCHA using the population activity of such neurons? What we found is very interesting. What we find basically is that if you take neurons where they multiply the responses to individual letters to get the combination of the response to multiple letters, all of these populations of neurons don't reach the level of perfect decoding. That is, that you cannot decode the identity of letters in a CAPTCHA if neurons were multiplying the information of individual letters. Okay? But if you take neurons that add the information of multiple letters, you can. And it turns out that there's very specific combinations of these rules which actually work. And specifically, exactly these two rules are the ones that you need to work. Okay? So if you want to get perfect CAPTCHA decoding, what you actually need to do is to multiply distortion and shape information and add multiple letter information, and these two rules are exactly the ones that you need to actually crack or perfectly decode CAPTCHAs. Okay, so that's interesting, that's very nice. So then what about deep networks, right? So maybe will something similar actually happen in deep networks? Okay, because in these cases these deep networks are actually trained for text recognition, and they're trained in a sort of supervised manner to just recognize the identity of letters in a string. And so the question is, do these rules automatically emerge out of the result of training? Barney, you had a question?
So, your multiple letter distortion, right? So each independent letter was distorted differently.
So we apply the same type of distortion to every one of them. Yeah. So because otherwise then every distortion is different, so we won't have any reason to expect this kind of separability. So it has to be the same distortion applied to multiple sort of units. Yeah. So then the nice thing is we have this analysis that we're doing on individual neurons. We can take the response of the neuron to multiple letters and distortions and multi-letter strings, and then perform the same analysis on deep networks also, because they also have neurons, they also have units that activate in response to letters, and you can just do the same analysis on deep networks. So this is the result. What we are doing is we've taken deep networks trained on a bunch of different ways, and what we are showing here is the responses to distorted letters, where you're showing every possible letters across every possible distortion and then fitting these models. And it turns out, if you notice over here, the M is the multiplicative model and the A is the additive model. It turns out that the multiplicative model is explaining the activations of the units in the deep network exactly better than the additive model. Okay, and if you look at the response of these networks to multiple letters, again here we find that the additive model is actually better. So it turns out that these deep networks are actually learning exactly the same rules that are there in the real biological neurons. That is, that they have to multiply information about shape and distortion and position as well, but they have to add information about multiple letters. And that's what gives you a nice invariant kind of letter code. If you look at CAPTCHA decoding itself, it turns out that the neurons that actually show very strong separability or strong differences between the shape, between the multiplicative and additive models, are actually the ones that do perfect CAPTCHA decoding. In other words, if the rule is present more strongly, then you get better decoding of CAPTCHAs in that network. And so it also shows that the presence of these rules are actually kind of important for getting good CAPTCHA decoding. Okay? So that's the second study. So if you're comparing machine and neural responses to CAPTCHAs, we can extract the rule from real brains. We can check if that rule is present in artificial vision algorithms, and then show that actually there are certain rules that you need to crack CAPTCHAs. Okay, so that's part one. Any questions? And I'm going to switch gears into a completely different thing. Okay, so we'll take silence as assent in the true Indian Yeah.
This might be silly, but how do you motivate the addition and the multiplication rules as the two choices you pick?
So, multiplication, I can tell you the intuition behind this is that, let's say you have two signals, right? If you add those two signals, then in some sense you're not going to be able to recover the individual signals from it, because if you have a large signal and a small signal, then you add the two and the identity of the small signal is gone. But if you take a multiplication, then you can preserve the identity of the small signal as well as the large signal. That's at least my intuition.
So you're modulating two sort of signals together.
That's right. That's right. So the idea is that if you want an invariant representation, maybe you don't need to achieve perfect invariance, like the response of the neuron is only there for the exact same response for all views of an object, but the view is modulating the neural response in a very tiny way. And that tiny modulation is a multiplication. That's at least the intuition. We're doing some experiments to check on some of those things, whether experience and familiarity modulate these rules or cause the emergence of these rules. But right now, that's what we have. We've also checked the responses of these neurons to multiple objects containing multiple parts, and this seems to be a fairly general rule that the response to multiple parts or multiple objects is usually a response is the average of the responses to the individual parts. Yeah.
So does the dynamics play any role, because your real response are going to change as a function of time, right?
Yes. So in the study we did on invariance, I didn't show you that study, but basically we looked at how IT responses to objects across multiple viewpoints or sizes or positions are modulated, and there we generally see a kind of more greater invariance in the later part of the response. But that's an effect which is there, and that could be because of just a weaker feed-forward computation or maybe recurrent process. We don't know. Yeah.
How to use this insight about comparing neural network and brain, like we say, global advantage to make that
better? The short answer is I actually don't know. As neuroscientists, we sort of grow up with querying neural systems and kind of extracting principles out of them. How to make those principles, how to sort of force these principles into neural networks is something that we're looking for people to sort of collaborate and tell us how these kinds of things can be done. But in general, I don't know what kinds of architectures or what kind of data sets are required to get, say, for example, a global advantage. There are some clues. For example, ImageNet, which is the most famous sort of image database, contains, for example, 40 or 50 types of dog breeds. Now, if you think about it, all dogs have sort of the same global shape, and maybe the local features of the dog breeds are different, and that's what you need to recognize those dog breeds. So maybe if the kind of training that is done to the network is changed in terms of requiring it to respond to more global categories, maybe it would acquire a more global sort of advantage, but we don't know.
Considering all those boundaries and parameters, can you say that network would be better in vision?
So what we can do is to say, okay, let's do whatever training and whatever optimization we want, but we can tell you that these are the properties you can check, and you can check whether these properties are present or not. Because the danger of these things, I think, I mean, this is again going into a little bit of, you've got to think about the philosophy of the approach, because if you optimize the very thing you want to get back, you will get it, but like you are not getting anything extra. So I think the interesting thing would be to train neural networks on some task where you have no reason to expect a global advantage, but then you get it now. And so then you can say, okay, maybe you need a global advantage to solve this task.
But then you already have the bias that you need global advantage.
Well, because brains are doing vision better than machines. So you got to carry some bias that brains know the solution, right? Okay, so I'm going to switch gears now and tell you about something that our lab has been quite busy with understanding over the last couple of years. And this is to try and understand the neural basis of real world vision. So what do I mean by that? Well, to understand what I mean by this question, what I want to highlight to you is that there's a particular approach that has been used for decades in trying to understand vision in the lab. And the traditional approach is basically you take the monkey, you take the animal in general, and you bring it into the lab, because you want to record very tiny electrical signals from the brain. You need to bring it into some sort of shielded, electrically shielded chamber. You need to pick up very tiny electrical signals. All kinds of extra stuff is there. So the approach actually that most labs or most places that have done this is basically you have an animal which is sort of tethered in place. Okay, it has to be tethered because you need to figure out where it's looking. It has to also be tethered because you need to record from the brain. And monkeys have hands, So if you put a wire sticking out of the brain, then monkey will just take it and pull it out. And that's the nightmare of most primate neurophysiologists. Okay, so now, and then you have also to give juice reward, and so the juice reward has to come out with something. So it's like a very, very constrained kind of situation. Okay, what you need to do to actually get a signal out of the monkey like this, and this is the kind of recording that I just showed you, the playing of with the viewpoints and the animals. So those recordings were done in this kind of setup, where the animal is brought into the lab, and then you're plugging in some electrode and plugging in the some amplifiers to record electrical signals, and then you're doing the analysis, right? What do you need to make all this happen? Well, you need to put a head post, so to stabilize the head to record. You need to train the animal on tasks. You got to implant the electrodes. You got to record the brain activity. But something is missing in this picture, which is that this approach is actually blinding us from knowing how the brain responds in the real world. Okay, because in the real world, the animal is free to move, and all kinds of extra information might be coming into the visual cortex, which is not being observed because the animal is in a very constrained situation. Okay, so the question is, can we actually free ourselves from this entire sort of very constrained setup? This is the sort of the dream that we've been pursuing over a couple of years. Some of the people in the team are actually also here in the audience. And this is the sort of the dream team. And the basic question we wanted to ask is, can I actually understand brain activity during natural vision? So for example, I'm not sitting in front of a screen. I'm going to pick up an object, and the object has some complex kind of image that is producing on my retina. And then I want to understand actually how to actually go about making sense of all that complexity. So let me start again from very basic first principles and say, is it enough to record brain activity in a natural setting, right? So this is, for example, the environment in which our animals live. These are no longer cages, by the way, because they're living in pretty nice large enclosures, probably bigger than like people, our own rooms are not that large, okay? The question though is, first of all, will it even work in a simple task, okay? The second is, will it work for complex sort of social interactions? And the third is, will it work for sleep? And I'm going to take you through like a kind of proof of concept that all of these things can work, okay? Why do I think that it even won't work? Okay, so we got to start and think about why is it that neuroscientists have been taking animals into a very, very constrained setting in the first place, okay? Yeah. So, well, if you look at a natural task, so I'm going to show you videos of a task where we've recorded brain activity. Basically, the task is very simple. And our animals love this task because this task is actually not even a task. The task involves the experimenter enters the room, comes in with a box of food, opens the box, and the animal has to reach into the box, take the food and eat it. That's it. Okay. In our other tasks, the animal has to do a lot of things to get reward. Here it's just like you're just taking food, right? But look at so what I'm going to show you now is actually videos of many trials of the same task where different people entered and gave different kinds of food. So there are many different experimenters and many different kinds of food, and all the trials of the experiment are being shown here to you, replayed at the same time. Wait, what's going on? Okay. So the animal is sitting here. We have CCTV cameras that are actually observing it. And then the experimenter has come there. The animal is coming up to receive the reward. You notice that he has a little bump on the head. And that bump is actually not because of a fight, but actually we've implanted a wireless transmitter to record brain signals from multiple regions in the brain, okay? The other thing that you might have noticed is that different trials are different in length because the experimenter comes to offer the food. The monkey may not be interested at that point in time or didn't notice the experimenter. He comes in a little bit later, okay? The monkey may not reach exactly at the same time. So there's all kinds of variability in this task, right? So if you start, if you talk to somebody and you say, okay, I'm going to record brain activity in a natural task where the animal is kind of coming in and I'm going to give it some reward. And remember, then immediately, and this is particularly true because, some people are very good at saying, telling you all the problems that could happen if you try something out, okay? So here are all the problems that you can get. So, for example, in a real-world task, there's lots of variability across trials. In a real-world task, you're giving a real stereo image, right? You're not showing stuff on a monitor. So the view that the stereo information extra is coming in. We also don't know where the animal is looking, because we can't track the eyes in a real world scenario. You can't implant an eye tracker, because eye tracker is an expensive piece of equipment. It needs infrared vision and all kinds of stuff. So then you don't know where the animal is looking, right? We also don't know the animal's own movements, because the animal could be moving and all that information that the animal is moving might be coming into visual cortex. So this entire scenario is actually kind of scary, right? So how do we actually make sense of it? Because in a natural task, there's so many different sources of variability, including our own, because you're actually coming in at a different rate and you might be reaching and interacting at a different rate. So then the solution that we've come up with is basically to say, okay, let's learn what we get with neural responses on a screen, and then you test it in a natural task. So you have kind of a rigorous and well-controlled environment where the animal can do a task. We can record responses to lots and lots of, showing an image lots and lots of times. But then in the test condition, you're just showing that image once, and you're just offering the food once to the animal and saying, okay, can I actually figure out which food was offered, which experimenter saw it, and so on. Let's just do some basic decoding, right? So how do we do it? Well, so that you can see the crux of this. You need to have on one end, you need to have a situation where there's a very controlled setup, and then on the other end you have the uncontrolled setup where there's a natural arena and so on. Okay, so the philosophy is basically now the reverse, which is that now instead of taking the monkey into lab, you're bringing the lab into wherever the monkey is, and that's a profound change in our kind of way we do the experiments. So in order to achieve that particular profound change, well, first of all, you can't have a head post. You can't restrain the head of the animal. You got to do this in unrestrained animals. They're moving freely. You got to train animals in a task on a natural setting. And then the other crucial thing is you need to record brain activity wirelessly. And this is not trivial at all, because we want to record huge amounts of data, and then that all the data has to be stored, synchronized wirelessly to the experiment computers and so on. So there's a lot of complexity to that. Okay, so here's how we did it. So this is sort of a bird's eye view of the entire the new lab that we built. So we actually had to build an entirely new lab to make this happen. So this is the final result. So the final result is, this is where they live. They don't live in cages anymore. Our animals live in nice, comfortable, sort of naturalistic environments. They commute from this side to a more controlled environment where they have to go through a sequence of doors, because we want them to live in a group, but we want to be able to separate the animal that we want and record from his brain. And so we open and close doors to induce the animal that we want, give him some raisins, give him some juice rewards or whatever to make him come to the experiment. Finally, this is where they come and work. So they come here, there's a touch screen, and then there's a juice spout. And so they sit in front of the touch screen, do some task and get juice reward. Okay. So this is looking very simple at the end of the day, but there's a lot of iterations that we have to do to make it happen, because there's a complexity to this particular place. Okay, but it's very similar to the kind of situation that we all have. We live in some place, we commute, we go to a touch screen, and then we get some juice reward when we drink coffee. So it's not too different from all of us. Okay. If you go to the juice spout, there's some complexity here, because there's a particular juice spout, and you see this is a 3D printed chin rest, which we actually printed based on the scanning the animal and to knowing the exact shape of his face. And the reason we actually printed the juice spout like this is that the animal can, when the animal wants juice, his head comes to an exactly stereotyped position. Okay, there's no variability in exactly where he puts his face, because there's only one position from which he can drink juice. He can't take the juice like this, because the chin rest will block it. He can't take juice like this, so he can't do any monkey stuff. He has to approach the juice spout directly and then take juice. Okay. What this allows is actually the ability to track his eyes, because the moment you bring his eyes into a stereotyped position, you can track the eyes. And this is what we have. We have an infrared camera that's shining light on the monkey. The monkey is sort of sitting in front of this juice spout, and then the reflected image is actually captured by cameras, and there's some complexity to sort of how you track the eyes, and we're able to get good eye tracking. Okay. So now the final sort of setup here. So you have the animal, we have the brain data being recorded wirelessly. One sort of little bit of detail is that the brain data is not being transmitted to our base stations. We're just synchronizing the data and saying that the experiment has started. We're sending some pulses between the computer and the wireless logger. The wireless logger is actually just storing all the data, because we're recording from 256 electrodes which are implanted into visual cortex and motor cortex. And each electrode is being sampled at 20 kilohertz. So there's lots of data being coming in. and even just 5 minutes of data is like 4 gigabytes of data. So there's a huge amount of data we are able to record in this setup. So what do we do with all this data? Well, you can take this data and you have the animal looking at images on the screen. And what we did in this experiment where I showed you the experimenter coming and giving stuff to the animal is that we actually showed pictures of that food box and pictures of the experimenter and pictures of the play area to the animal on the screen. Okay? So what you can then do is we can say, okay, I have some brain activity which is evoked by looking at pictures on the screen, and using that brain activity I can build a classifier which tells me which food is he looking at or which experimenter is he looking at. Okay, and so that's what we do. We train a linear decoder using responses to images. Here we can present the images any number of times because this is an experiment where lots and lots of trials can be made. He's getting juice for every time he does it, so he's happy and we are happy. Okay. Then the test condition is to eventually test this decoder in a natural task, because in the natural task we have only a couple of trials. We can't keep offering food. He gets full after some time of eating food, so a very limited opportunity to actually record brain data. Okay, but before you go to this level, you want to do a sanity check, right? The sanity check is simply this: we just want to test the decoder on new trials where we've never seen this neural activity before, but we still want to check whether the classifier is working, and that's exactly what we did here. So this is all responses to neural responses on the screen, but on new trials. So basically this is the cross-validated decoding accuracy, and we're able to decode the identity of the food by looking at the neural activity in the visual cortex. We're also able to decode the identity of the experimenters. So these are 3 people who've entered into the room during that experiment. Turns out actually in this case, only these 2 people were actually giving food to the animal. This experimenter was not even there in the room. Okay, so this is sort of like a control where we're just looking to see that if we train a decoder, the decoder should never say that Cine is actually giving the food. It should only choose between these 2. Okay, but in the screen task, we actually showed all 3 images. All right, so we are able to decode the identity of the experimenters pretty well. And so now the stage is set and we're able to record, we can correctly guess which image is being seen by looking at only the neural activity. So that's the key, right? So you're looking at neural activity and we can tell which image is being seen. So now how does it work out in the natural task? So we have the same setup here, and you can see here that this is actually now decoding in the natural task. The red bars indicate the upper limit which is obtained from the screen task, and in the natural task we can see that we are able to decode the identity of the food being offered above chance. We can also decode the identity of the experimenter, but this decoding is not very strong. And that makes sense because the animal doesn't care about the experimenter giving the food, he cares about the food. And so the food is being decoded much better than chance. Okay? So we're able to correctly guess the identity of the food, the experimenter. And actually, by the way, we're also recording from motor cortex, so we can actually tell whether the animal is using his left hand or the right hand, and we can decode the hand that the animal is using in the same task in the same way. Okay, because we made the animal take the food with his left hand, we also made him take the food with the right hand, and then we can, based on the pattern of neural activity, we can tell that the animal is using the left hand or the right hand. Okay, so we have basically proof of concept saying that in a simple task, we can go from the screen, that is you can train decoders on the screen, and then you can test it in a natural setting and you can decode. Well, this is only the beginning. And we want to do a lot of different types of things. So the next question is, will it work in a complex social interaction? And actually, I think this might be the first time that anyone has recorded simultaneously from the brain of 2 animals socially interacting, at least for monkeys. So we've managed to do that. So this is sort of a screenshot of the entire trial. Basically, what happens when 2 monkeys are introduced into the same arena? What do they do to each other? Well, they groom each other. Right? You must have seen this if you have looked at all the monkeys on campus. They will groom each other. The scary thing is that they'll fight each other, and the fights are actually horrible and deadly. So we had to do a lot of things to make sure that the 2 animals are compatible and they won't fight and things like that. But in this particular task, they did not fight. They were just grooming each other. And so we're able to record neural activity while they are grooming each other. Okay? They're both male, yeah. Yes. But monkeys of all types have all kinds of fights, and it's kind of incredible. They can really damage each other, and it's in ways that you would not expect, but it happens. Yeah.
quick question. Were there any cases where the monkeys were trying to rip off the head stage? Because we did something like this for pigs. The first thing the other pigs would do is like, very inquisitive. Like try to
right. So there's a, so there's a kind of insider trick that I'll tell you about monkeys. If they have an implant on the head. So obviously they know that there's an implant on the head, they can touch it. If there's any kind of mechanical wiggle, then the animals just go completely after it and they'll kill your implant. But if there's no mechanical wiggle and doesn't rattle or do anything, they'll leave it alone. This is exactly like children, if they're doing, if there's something to rattle, they'll rattle it. And that's it. So they're actually okay. But this is a huge point because there is a kind of non-trivial amount of time to figure out a good implant. And we had to do a lot of iterations to actually get there.
The wireless head stage is something that you guys totally did in-house.
Yeah, so we 3D, the base of the implant actually is titanium because it has to be biocompatible. And so the base of the implant was 3D printed in titanium and we designed it in-house and then we printed it locally and then we had to 3D print the cap. We had to make sure that there's no liquid that gets into the electronics because there's a bunch of electronics that live on the implant now. Yeah. And so there's a lot of stuff maybe better told over beer than in a talk. Okay, but there's a lot of iterations that we had to do to make it happen. This is what I'm showing you now is actually each row is the activity of one channel. So I'm showing you spiking activity and we're recording from 2 brain regions in one monkey. This is IT and premotor cortex, and 3 brain regions in the other monkey, IT, premotor cortex and prefrontal cortex. And these are all the brain data that we've recorded in the entire session. This is just to give you a sense of like, this is the complexity of the data. So what do we even want to do with this data, right? So one question is, can I actually decode whether the animals are grooming each other? And remember there's 3 types of groom events. One is that the animals may not be grooming each other. They might be just sitting around and looking around at our beautiful recording room. But they could be grooming each other. And it's very interesting if you've noticed this legend over here, they actually sometimes the M1, monkey 1 will groom monkey 2 and then he stops and then monkey 2 will groom monkey 1. And if you look at the total amount of time that they've groomed each other, surprisingly it's actually almost the same. Okay, so it means that they're keeping track of like how long they've groomed each other and kind of returning the favor. Okay, it's pretty complex and they're kind of it's a complex interaction, right? So the question then is, can I actually decode whether the animal is being groomed or is he grooming somebody or is he not grooming at all? So those are the 3 states, and again we can build a classifier, and so we built a classifier to check whether the animal, so this is from monkey 1, I'm reporting the decoding accuracy of our ability to use the neural activity to infer the grooming state. And it turns out that we can, not very surprising because the brain is controlling the grooming activity, so this is again still proof of principle. The second question we wanted to know is, well, in a grooming situation, is the animal keeping track of his partner's identity? Does he know who he is grooming or who's in the room with him? And so to answer that, what we did was to present pictures of him and his friends on the screen, train decoders on those images, and then test those decoders in the natural task. And so this is the decoding. I'll get to you in a second, just give me a second. So then these are the decoding probabilities of actually from monkey 1 and monkey 2. Basically what we're saying is we're able to decode who the partner is. That is, from monkey 1, I can build a decoder based on monkey 1, 2 and 3. And if I'm recording from the brain of monkey 1, then the decoder tells me that monkey 2 is in the room. And if you're recording from the brain of monkey 2, then the decoder tells me that monkey 1 is in the room and not monkey 3. So basically this is all very sensible kinds of decoding that we're getting. Question?
The classifier, it's trained on each monkey.
Yeah,
right?
That's right. That's right. The signals are different and the selectivities of the channels are also different. So we may not be recording from a face neuron in one monkey in the same channel as the face neuron in some other monkey. Yeah. Okay? Yeah.
These experiments were done over many days, right? Or just 1 day?
This is actually a 10 minutes or 15 minute session of social interaction.
Yeah, so I was going to say that decoders are probably going to change from day to day and so on.
We've checked this. The neural selectivity that we get on a particular day sort of doesn't change too much across a couple of days, but over a time scale of maybe about 30 days or so, there is a change. And we are doing a little bit of analysis to check whether there is any kind of change in the selectivity across days. But usually every experiment is done on a particular day, and that one day of recording or even that 1 hour of recording is sufficient for us to keep us busy for hopefully not too long, but certainly for weeks and months. Yeah.
So you need to train the decoder again, so over a long period?
No, so in all these experiments what we do is we do the screen task first. So the animal does some task on the screen, then he does a task in the natural setting. And so we're always building the decoder on the trained data on the same day, because we don't want to necessarily worry about the selectivity changing and the decoder becoming worse across days. Yeah.
So here you're recording from, you're building classifiers of 2 different monkeys. But
that's right.
Are they in agreement at the periods? So what I mean by that is,
so we have,
and one is moving M2.
Right. So we've checked. Yeah, we've checked these things, and what we actually find is there's not much difference in the strength of the decoding between the different groom bouts, which seems to suggest to us that they're just keeping track of their social partner throughout the session. They're not really it's not dependent on when the animal is actually directly looking at that animal, at his partner.
I see, but when you classify correctly, for example, is the monkey 1 correct classification temporarily correlated with monkey 2's correct classification?
We have not checked that particular point. I don't think we saw very clear patterns, but we've not checked it in terms of whether the decoding itself is correlated. Right. What we are doing nowadays, more recently, is to check whether the brain activity is getting synchronized over the course of a social interaction. This is something that's been observed in EEG recordings from humans. But we're checking this at the single neuron level, and hopefully some interesting insights would come out. So this is sort of ongoing work.
That might correlate.
Right. It's an interesting idea. We haven't really thought about it. I don't think we noticed, at least at the level of the groom bouts. So for example, if you look at the decoding of the faces so for example, you could speculate that when monkey 1 is grooming monkey 2, he's looking at the face of monkey 2, but it's not actually all the time, because sometimes he's looking at the fur and sometimes he's looking at the back of the body, and so we don't know. But even on periods when he's seemingly directly looking at the other monkey, we don't see necessarily better decoding. So what we're taking this to interpret is, we don't have very good eye tracking, but at least the periods where in the video we can feel like the animals are looking at each other, that doesn't seem to change it too much. Yeah, but it seems to suggest that over the entire grooming session, they're just keeping track of who's in the room. Because I can look at, for example, the decoding of a third monkey, which is not even present in the room, that decoding is always very low. So we know that he's keeping track of like the monkey actually in the room. Yeah. There was some hand back there. Yeah.
Just a quick question again. How many pairs have you tested this on?
How many pairs of animals?
Yeah.
This is just 2 animals. So the story in monkey neuroscience is, if you got results from 1 monkey, it's an anecdote. But if you got data from 2 monkeys, it's gospel truth. So that's it. This is it. So, but these are actually long and laborious experiments, and that's why the results, that's why it's the common practice in our field is that you just record from whatever results you get from one animal are expected to be replicated in the other. But usually there's just too tedious to conduct these studies over many more than 2 animals. Yeah.
How do you distinguish between self and other? Like, is self considered like a new face that the monkey?
Yeah. So what we did here was actually we showed photographs of. Let's say if you take monkey 1 is shown pictures of monkey 1 himself, also monkey 2 as well as monkey 3. And so we can build a decoder now by looking at the images of monkey 1, monkey 2, monkey 3, and then look at whether we're able to decode in the social session whether he's looking at monkey 1, monkey 2, or monkey 3. And turns out that if if you're recording from monkey 1's brain, then the decoder corresponding to monkey 2 is active, during that session, but not monkey 1. So I mean, they have not seen themselves. But occasionally they will see themselves in the touchscreen reflection, and then they'll kind of freak out thinking it's another monkey. So we don't know we've never recorded that data, so we don't know.
Yeah. So according to this data, monkey 1 doesn't really care who he's giving grooming to. Could be a partner, could be other.
Oh, in this, usually the partner is decoded during different times. That's right. That's right. He doesn't care. Some monkey grooming me. Yeah. So possibly, and these are things that we are a little careful to not over-interpret, because we are just looking at decoding based on looking at the screen task, and potentially the responses in the real world is not going to be the same as just taking pictures and doing it. But we are able to generalize and get sort of sensible generalizations in some cases. Yeah.
So you have recorded from monkeys in the traditional way except for data.
Right.
And now this data is new in the field.
Right.
Does the data look the same, different dynamics, the neural responses?
It's a great question. So we thought, when we were starting on these studies, one thought was maybe we kept the old lab as backup, thinking that if everything fails here, like we'll go back and record in the old lab. And so that was sort of the idea, that we'd be able to record in the old lab and sort of do a very constrained kind of setup, and then do it in this new setup. But after some bit of thought, what we realized is that that's not going to be a meaningful comparison necessarily, because there's so many things different between that setup and this one. So I'm not sure that we can actually be able to go back between the traditional setup where it's completely head fixed recordings. What we can do is to be able to record in the neural activity in the screen over here, and then see to what extent that generalizes to the natural setting. So I think it's a bit early to say whether we are seeing something dramatically different in the natural setting. What we are looking for is very specific types of information. So for example, one question is, how much does the movement of the monkey actually reach the visual cortex? So that is, is the visual cortex getting information about the movement of the animal, which you would not even know in the traditional setup, because the animal is not even moving, right? Similarly, how much visual information is coming into the motor cortex? Similarly, like when you're interacting with a real world object, then how are you manipulating? What's the visual cortex doing? What's the motor cortex doing? So I think the nature of the questions are different, and it's a bit early to say whether we are seeing something dramatically different between the constrained setting and the real world settings. And I think, my guess is that a bunch of things will generalize into the real world, but there may be some new information that, of course, would be the interesting part. Yeah. Final bit, I don't know how much time I have, and I don't know if I've gone over time. Okay. So final bit, and actually this is, I can promise you it's going to be interesting, is that I've shown you social interactions, and this is sort of very exciting for us, because question is, can we say something about sleep? And this is very unique and different, because again, in the traditional setup, the animal is brought into a lab, he's definitely not sleeping. And he's definitely not sleeping when we want him to sleep, right? Because so in this real world, kind of in a very naturalistic setup like this, we can actually record when the animal is sleeping, because he is sleeping in the same area, and I can just record when he's sleeping. And so the question is, what can we actually say about neural activity during sleep? Again, very, very little work that has been done in terms of looking at primate brain activity during sleep, because primates just don't sleep in that constraint setup, right? So here's the bottom line. So I'm just showing you like two screenshots of a CCTV video where we're monitoring using infrared CCTV. So the animal is actually in a room with no lights, and what you can see here is the eyes of the animal are not like that. This is the reflection of the infrared light from the animal's eyes, and so that's why his eyes are looking white. Okay. So we know the animal is sleeping because we know that his head is down, and monkeys, by the way, don't sleep like on the floor sprawled out like us. They actually sit and sleep. They hunch down and sleep. Okay, that's how they sleep. I didn't know that until we saw them sleeping here, even though we've been recording from monkeys, for a while. So here is now sort of the recording during sleep. What we've done is to annotate this video to say that when the animal is actually awake, and just like us, they kind of wake up in the middle of the night, move around, scratch themselves, do a bunch of stuff, and then go to sleep again. So we annotated every one of these bouts, saying that, okay, this is when he's awake, this is when he's sleeping. So then what the hell are you going to do with all that annotation? Well, the very basic question we wanted to know is, from the neural activity, can I tell if the animal is sleeping? Right? Basic question. Again, it's a decoding question. You build a classifier, you can train the classifier on these annotated bouts, then you leave aside some bouts which are actually left aside, the model has never seen it, and the question is, can you tell whether the animal is sleeping or awake? And so it turns out that you can. And this is the result that we have. So this is the accuracy of decoding. What is also interesting is we can predict if the animal is going to sleep. And so all you have to do is to actually now run the decoder, but look at the transition points where the animal actually went to sleep or woke up. And turns out that actually I can predict this is the average decoding, this is the decoder probability. And you can see that when he's going to go to sleep, you can see the decoder kind of starting to dip down. And when he's going to wake up, you can see the decoder starting to kind of dip up. And so what we know is that now there's some kind of anticipatory, or there's an advanced sort of notice that, okay, the animal is going to sleep. And again, I still think this is all proof of concept, okay? It's not surprising, in other words, that if you're monitoring brain activity and the brain is the one that actually makes you sleep, so there's not surprising that you can do all of these things. But it's interesting that you can, because even getting to this point and getting this kind of data is not easy. Yeah.
Can we decode different sleep states?
That is exactly the next slide, okay? So then the question is, can I define a kind of dream state? Okay, now this is a little bit complicated for us because normally if you want to define a dream state, it's that you need to have rapid eye movements. And we're not recording eye movements in the animals, so then how can I define a dream state? Well, we thought, okay, let's do some bunch of analysis and try to think of some sensible ways of defining the dream state. This is what we came up with, okay? What we do is we actually take the average brain activity during the entire wake period and the entire sleep period. And then we say, on a running basis, can I calculate the similarity between the current brain activity to the sleep template and the wake template? That is a template of activity or the average activity that I get during the sleep state and the wake state. So then what we said is, well, there are periods during the time when the animal is awake where the similarity to wake is very high. That's of course the wake state. That's not surprising. But there are actually periods in the brain where the animal is actually sleeping, which is the blue shaded region. We know the animal is sleeping because we've annotated the video, we've looked at the animal sleeping. But those periods, the similarity to the waking activity is actually high. Okay, it's as if the brain is awake, but the animal is sleeping. Okay, so that's what we thought, okay, maybe that's sort of REM sleep. Okay, well, we have more than that, because what we can do is during the fixation task, or where the animal is actually doing a task on the screen, on the same day where the animal slept, during the day we made him do a fixation task. In the fixation task, we showed him pictures of food and we showed him pictures of empty food boxes, because that was the experiment on that day. We showed him food boxes, he was given food and not food. We can now train decoders on the neural responses in the screen task and then test it during these periods, right? And we can say, okay, if the animal is sort of dreaming about food, then the decoder corresponding to the food will actually be active, right? Sensible sort of expectation.
He's not going to dream of empty food boxes.
He's going to dream about food. Okay, so this is the classifier that we built. The classifier simply says, is there food in the box or not food in the box? Okay, so this is the classifier learning. And what you can see here is very interesting, which is that if you compare it to the non-REM portion of sleep, that is the same sleep period, the non-REM portion, the decoder is actually more active in the REM period than in the non-REM period. Okay, and that tells us that maybe during the REM period his sort of activity is similar. Also notice that in the awake state, because his eyes are open and he's not looking at food boxes, the decoder is actually kind of low. And so this is also kind of sensible result to us. Remember, I must say some caution here, because we are interpreting the activity of a decoder. We don't know what the animal actually saw or what he actually dreamt about. Okay, so there's a lot of qualification to this kind of result, but still it's interesting that we get anything in the desired or interesting directions. Yeah, Ernst.
Yes, so activity is lower, but it's still 50% probability that he thinks of food.
So that's right. So it's I don't know what chance is. I guess chance is 50%. So there's kind of slightly more than chance, I think. Yeah. So here the decoder is basically trained between like a bunch of images of food in food boxes and and empty boxes. So basically the decoder is just saying, is there food or not?
I see. So
Yeah.
there's many other things they could think of. So
That's right. So that's absolutely right. So for example, we don't know what else he may be thinking of, and actually it shouldn't be the case that he just thought about, like, I don't know, he just thought about me, and then it looks like the decoder is just projecting that into like a food versus non-food. So yeah, we don't know these things, and I think, but I can say one other thing which is kind of interesting and addresses maybe some point. We also can train the decoder on faces versus objects. And we can say, okay, during the different phases, is there some differential activity in the decoder corresponding to faces versus objects? And there isn't any. The only place where we see a difference in these in any decoder is actually in the food decoder. So in that sense also it kind of matches with the idea that maybe the animal is sort of thinking about some kind of food. But again, I do want to qualify that that's not 100% the case, because we are only classifying between 2 or 3 possibilities.
So how do we get the drawn to? How do we know that the animal is doing this particular thing?
That's again a question that we can chat about, like over not in a talk, but over some kind of suitable juices. Yeah.
So is it possible to classify beyond red states, like in a non-red states, like deep sleep or something like that?
So typically in the sleep literature, what people do is to monitor the eye movements, and we don't have access to the eye movements in the animals here. We've tried to kind of build like decoders of eye positions using the neural activity. It's to cut a long story short. Right now, we don't have any good way of knowing what the eye activity is like. And usually in the sleep literature, that's considered to be the gold standard. Like for example, if you're sleeping and your eyes are moving very rapidly, it's an indication that you're going through a dream state. And this has been confirmed by, if you wake up a person, like this is a good experiment to try on your roommates. That's what roommates are for. So if your roommates are if you see them sort of moving their eyes very rapidly in sleep, wake them up and ask them what they're dreaming about. In all likelihood, they're dreaming about something. If you wake them up during like a non-REM portion, they won't be dreaming about anything. Okay, so this kind of stuff has been done on human subjects. With animals, of course, you can't wake them up and ask them what they're dreaming about, because they won't tell you. Yeah.
So at least we can get the drawn to from the human subject, right?
That's right. This is there in the sleep literature in humans. Yeah.
Probably correlate the
That's right. So such things have been done, but again, not directly. In monkeys, it's a little bit harder because you don't know what they're actually going through and you can't ask them. Yeah. Okay.
So like decoding the monkey, what monkey is thinking with 100% probability, right?
Sorry?
If you're able to like decode 100%.
Yeah.
Get the total, it's essentially like decoding the monkey, what it's thinking,
right? I mean, already to an extent, I've already shown you that if you're choosing between a set of limited alternatives, I can tell you which alternative the animal is looking at or thinking about. Thinking about is a little harder because we don't know at what point he's thinking about something versus like actually looking at something. Okay, but the sleep is one state where you can kind of think about what could be happening. There are other situations where, for example, the animal has to hold something in memory and then act upon it. And those situations also we can say, okay, the animal must be thinking about it, because if he didn't think about it, he's not going to be able to solve the task. And those situations we've also recorded brain activity, and we do have brain activity trying in those periods where we can say, well, is the animal holding something in memory, right? Okay, so just to, yeah.
Like, how do you classify the non-dream and dream state by recording?
Basically what we did is we look at the average activity in the brain during the wake state and during the sleep state. And for any particular time instant, we just say, is it closer to the dream state or to the wake state? And then we already know that he's sleeping or dreaming, so sleeping or awake. So you take the sleep portion and you say, during the time he's sleeping, is there a time when the activity is very close to the wake state? And those periods we're defining as kind of like, sort of you're awake even though you're actually sleeping. And that's what we define as the REM state. I mean, this is not like that's not the gold standard, but like it's some sensible choice we could take and do something with it. Yeah. Okay, I think I should, yeah.
Is it not possible to record the eye movements using some bioelectron system?
It is, but it's just not something that, I mean, we sort of went through a lot of sort of trials and everything to get to this point even, so that's probably a thing that we might include in a later kind of recording. But yeah, it should be possible, but it involves extra sort of implantation, and you need to actually monitor the eye muscles. That's not trivial because you need to implant something here or near the eye and so on. And the animals might actually be able to, take it out, and it's a whole other kind of complexity to actually achieve it. Saying it is very easy, but I mean, kind of getting it to actually work is not trivial. So we decided not to go into that. Yeah. Yeah.
Is it possible that during the head down phase, the monkey is actually not sleeping and just keeping its head down?
Yeah. So we don't have additional confirmation that he's actually sleeping. I mean, but we've watched, I mean, I think collectively in the lab we've watched enough animals sleeping to know that they're sleeping. But again, like there's no 100%. I mean, so like I said, the 100% confirmation usually comes from like these extra signals that people record usually. But we're fairly confident in our assessment that they are indeed sleeping because they remain motionless for a long period of time. Their behavior in terms of waking and sleeping is very similar to ours. So we know that they're actually they are indeed sleeping. Yeah. Okay, so final thing. So I guess I've shown you all of this. So I should just wrap up. So I've shown you two parts. So one part, I've told you that machine vision algorithms are very different from brains. There's a bunch of similarities and a bunch of differences, so worth investigating further. The second part, I've also told you about natural vision, and brain activity is kind of scary to study in the natural world, but then that is ultimately, as neuroscientists, what we want to study, to go into the natural, sort of looking at brain activity in the real world. And so that's what we are doing right now. And this is our lab. If you're interested in talking more, or interested in more of what we do, please do get in touch. And these are all the awesome people that make all this happen. And thank you.